I'm pleased to introduce Dr. Grzegorz Ira. He's a professor of molecular and human genetics and a member of the Duncan Comprehensive Cancer Center. Dr. Ira is a well-recognized expert in DNA repair and recombination. He is being recognized for his novel insights into genome instability and the role that instability plays in homeostasis and evolution. This work has broad implications for cancer and cancer treatment. Congratulations to Dr. Ira. I invite you to learn more in this video presentation on his recently published work.
Good morning. First, I would like to thank two people that are present on this picture. In some way, I am here because of these two people. First, of course, is Michael DeBakey. I am really thankful and honored to receive his award, the award after his name, one of the greatest surgeons and doctors in the history. But also I am thankful to the person that is sitting in a pajama, who is the former president of Poland and the leader of Solidarity movement. At the same time, he was one of the last patients that was seen by Dr. DeBakey. Lech Wałęsa was the leader of Solidarity movement that ended up the communistic regime back in Eastern Europe and opened the borders for people like me to get the education and job in foreign countries. So I'm thankful to these two people. I also want to say thank you to Brendan Lee for the nomination and the selection committee for selecting me for this award. So the title of my short presentation is Extrachromosomal DNA Fragments Formation, Mechanisms, Sources, and Consequences. So everybody knows that normally DNA is within the chromosomes in the nucleus and within this short circular mitochondrial genome within the mitochondria, about 500 copies in human or about 50 in model organism yeast. So occasionally, cells acquire extrachromosomal pieces of DNA. And here I listed few sources of this DNA. So DNA can come from pathogens, as shown here on the top left corner, from bacteria or viruses, but also DNA can be transferred from mitochondria to cytoplasm if the mitochondria are damaged. And if there is a lot of DNA damage within the nuclear chromosomes, also some short fragments can leak through the nuclear pores to the cytoplasm. Finally, retrotransposon cDNA or DNA from dead cells can be transferred to the cytoplasm. So what happens if this type of DNA is present in the cytoplasm? In higher organisms, such as humans, innate immune response is being activated, and this is very beneficial for cells because some nucleases get activated and degrade this DNA, and some cells that are attacked by pathogens are getting eliminated. However, if there is constant leak of short fragments from either nucleus or from mitochondria or from cDNA of the retrotransposons, there could be constant activation of innate immune response leading to chronic inflammation and some severe diseases. And here I listed two diseases that were recently described that are induced by short DNA fragments in a cytoplasm. So whenever the mitochondria are defective and or damaged, and there is no proper mitophagy that eliminates these damaged mitochondria, mitochondrial DNA leaks to the cytoplasm. The innate immune response is activated, leading to chronic inflammation and some severe diseases such as heart failure or Parkinson's disease. The other consequence of the presence of this type of DNA that is perhaps not much studied is that these fragments of DNA can be inserted at chromosomal breaks. So whenever the DNA and whatever is the source of this DNA, whenever this DNA is present in the cytoplasm, it can go to the nucleus and be inserted at chromosomal breaks as shown here. So we have decided to use this feature that this DNA, free DNA, can be inserted at chromosomal breaks and screen for mutants that have increased level of free DNA in the cytoplasm, because the outcome of this would be that these fragments will be inserted at chromosomal breaks. So the way that we did it, we induced a single break per genome in model organisms of yeast with HO endonuclease, and looked for the mutants that would have the higher level of insertions of short fragments during non-homologous enjoining. Previously, it was shown by several labs that small fragments can be inserted at chromosomal breaks. About 20 years ago, free reports showed that pieces of retrotransposons, cDNA of these retrotransposons, can be inserted at the breaks, and also mitochondrial DNA can be inserted at the breaks. And more recently, Lanzevakia lab showed that during VDJ recombination and a class switch recombination, there are insertions of chromosomal fragments of pieces of the genes at the VDJ locus that leads to additional diversification of the antibodies. And at least in one case, they proved that such antibodies can provide increased response to Plasmodium-infected erythrocytes. So this is beneficial for some population of India because they are resistant to malaria. So occasionally insertions can be beneficial, but most of the time the insertions would disrupt the genes and of course would have a negative impact on genome stability. So going back to our screen, we looked for mutants that would have higher level of insertions. And here what is shown is that in wild-type cells, we do not observe any insertions. When we amplify this fragment, this locus that was repaired by non-homologous enjoining, we don't see any fragments being inserted at chromosomal breaks. We screened about 2,000 of these events, and there is not a single insertion. However, when we moved to one of the mutants that we tested, named DNA2 encodes the protein that is nuclease and the helicase, very important for DNA replication and DNA repair. We observed dramatic increase of this type of insertions. We observed 8% of non-homologous enjoining products that are associated with a change in a sequence that was cut by endonuclease, carried these pieces of DNA. This is, to our knowledge, the first mutant that shows this type of phenotype ever reported. So when we looked at what DNA is being inserted at chromosomal breaks, and initially we expected that it could be mitochondrial DNA or pieces of retrotransposons, we were surprised that most of these inserted fragments are coming from the genome, from genes throughout all chromosomes in yeast. About 92% are coming from other chromosomes. And the remaining 8% are represented by retrotransposons. Here are some features of the inserted DNA at chromosomal breaks in DNA2 cells. We see that these insertions have the size of about 100 to 500 base pairs. However, occasionally we see shorter or longer insertions. These are copy-paste type of insertions, so inserted fragments are not lost from the original locus. So insertion leads here to gene duplication. And most of the time we see that the single fragment being inserted at chromosomal breaks. However, occasionally we see that multiple fragments get stitched together at chromosomal breaks. When we look more carefully at these insertions, we see that the inserted DNA is not random from all chromosomes, but rather there are some hotspot donors for the inserted DNA. And the largest hotspot is within the rDNA locus. rDNA in yeast is located on chromosome 12. There are about 150 to 200 copies of rDNA genes. But we see also insertions, hotspots of insertions throughout the genome. We see that they are often coming from the centromeres, from telomeres, and other regions of the chromosomes. So when we compare the inserted DNA to all different features within the chromosomes, we noticed that inserted DNA is often coming from the origins of replication, from centromeres, telomeres, and R loops. And interestingly, these last three features, centromeres, R loops, and telomeres, we know that DNA2 is very important for the replication through these regions of the genomes, because these are hard to replicate parts of the genome, and we know that DNA2, from the work of other people, that DNA2 is responsible for the replication through these sequences. Moreover, when we zoomed into single repeat of rDNA and looked carefully where the inserted fragments are coming from, we found that they are coming mostly from this region that is between the origin of replication and replication fork blocking sequence. So this fork blocking sequence is essential here to prevent collision between the replication fork and transcription bubble coming in the opposite direction. And we know that the DNA2 is essential for transcription through this region, and what it does there, it processes the reversed forks that are often formed at this particular region. So this observation, as well as the observation that the insertions are coming from the centromeres, telomeres, and R loops, led us to conclude that very likely the inserted DNA comes from the structures that are not processed properly in the absence of DNA2. So what we know about DNA2 is that it processes reversed forks, as it is shown here. And this is not just our imagination. These structures were shown by electron microscopy in the absence of DNA2. We see a lot of reversed forks. We propose that these reversed forks that represent over-replicated pieces of the genome can be released in alternative way and further pasted at chromosomal breaks. We provide some evidence for that, but I don't have time here to go into the details. Second function of DNA2 is to remove the long 5' flaps, and again, in the absence of DNA2, you would end up with a lot of these type of structures. We believe that these structures are trimmed by different enzymes, and we identified these enzymes, but I don't have time to go into this. This alternative cleavage of these sequences leads to their release, and these fragments, we believe, are pasted into chromosomal breaks. So altogether, we propose that over-replicated pieces of the genome in the absence of DNA2 can get pasted at chromosomal breaks. So this work was published recently, and we are happy that we have the follow-up story where we, in the collaboration with Dr. Guang Peng at MD Anderson, we show that innate immune response is highly activated in human cells after depletion of DNA2. So we believe that these fragments that are left unprocessed in DNA2 can be released eventually by alternative nucleases, and they will activate innate immune response in higher organisms. So with these results, we were convinced that we can screen for mutants that have increased level of insertions, and at the same time, we hope that we can identify enzymes that, or mutants that have increased level of innate immune response. So we decided to screen many more genes that are involved in DNA repair and checkpoint, but we knew at this moment that we cannot do it using Sanger sequencing because it's very time consuming. All these experiments that I presented so far were done using Sanger sequencing, so we switched to amplicon sequencing using MySeq platform. So the way that we do it now, we combine about 100,000 non-homologous adjoining products, isolate the DNA, amplify, as it is shown here, amplify damaged and repaired locus, and subjected to deep sequencing. And moreover, we can do this trick with many mutants at the same time, up to 20 to 30 mutants per single MiSeq run. We started with repeating our experiments that we have done with Sanger sequencing. So we basically did this experiment with wild-type cells and DNA2 cells, and here's the PCR product that corresponds to DNA repair locus. And we see immediately that in DNA2 minus cells we have a lot of inserted fragments, as opposed to wild-type cells where we don't see any insertions. However, some of them can be hiding and basically are too weak to see here. So here are the results of the amplicon sequencing, and we see only 4 insertions present in wild-type cells, and all of them are coming from the TY retrotransposon cDNA. And in DNA2 there is a dramatic increase of insertions, over 500-fold increase compared to wild type. And importantly, the distribution of the inserted DNA is very much comparable to what we obtained using Sanger sequencing. This means that we can truly use this method to identify new mutants with high level of insertions. So far we screened about 30 mutants, and 8 of them show significant increase of insertions. Some of them have dramatic increase of insertions, but it's too early to present it. Here I will now just describe one condition where we see an increase of insertions, and one of the first experiments that we did using this amplicon sequencing. So yeast can be used to study aging, because yeast divide about 20 to 30 times before they die, and one can isolate the cells that are old mother cells and have this type of scars indicating how many cells, how many times this mother cell divided. So we can isolate this population of the old cells and ask whether, in this case, whether there is increased level of insertions in old cells. So the reason why we decided to check the insertions in old cells is because there is dramatic increase of rDNA instability and formation of rDNA circles in old cells. Secondly, there is dramatic increase of damage in mitochondria. There is increased transcription and mobility of the transposons. And finally, in mammalian system, there is a low-grade chronic inflammation indicating that there might be some DNA fragments present in the cytoplasm. So here are the results of the amplicon sequencing from old cells. We see dramatic increase of insertions. For the very first time, this is the only situation where we see the transfer of mitochondrial DNA to the nucleus. We see a lot of insertions from mitochondrial DNA, but majority of the insertions are coming from this unstable rDNA. And when we zoom into the single rDNA repeat, we see something very interesting, that most of the fragments this time come from the transcribed part of the rDNA repeat, right from the 18S gene. We are very much interested to find out why these fragments are released only in old cells. I will just remind you that what we observed in the absence of DNA2, that all insertions, or most of them, were coming from this region between origin of replication and replication fork blocking sequence. So the pattern of insertions from rDNA looks very different in old cells, and we are aiming to understand why this DNA is released in such a large amount in old cells. So in a summary, I would like to conclude that there are several pathways leading to the formation of small DNA fragments, and at the same time likely to the activation of innate immune response. As I presented here, over-replicated and not degraded fragments of DNA in DNA2 nuclease mutants are being pasted into chromosomal breaks, at the same time, in human cells, there is activation of innate immune response. And in all cells, these fragments are generated from unstable rDNA, damaged mitochondria, and probably overproduction of retrotransposons. However, we see also release of the nuclear genome fragments. And we believe that all these four lead to the chronic inflammation in aging cells. So we are in the very beginning of studying these processes, and we hope to identify many more mutants that likely will be relevant to human diseases. So all this work was done by, or most of it was done by talented postdoc in our lab, Yang Yu, with some help for New Farm. And we collaborated with bioinformatician lab Kai-Fu Chen and Houston Methodist Hospital. We also got some help from Wei Dang, who help us to isolate old cells, old yeast cells. And we try to translate whatever we find in yeast into a human system by collaboration with Dr. Guang Peng at MD Anderson. Thank you so much for listening.